Beneath the surface of every pond, every ocean, every drop of water on Earth, something is spinning. Right now, as you sit still, trillions of microscopic motors are running inside you, in your lungs, in your brain, in your throat. They're beating in perfect rhythm, coordinating without commands, moving things you'll never see. They've been running since the moment you were born, and they haven't stopped once. These aren't machines in the way you think of machines. They're molecules, proteins folded into shapes that can spin, bend, and beat. They're 50,000 times smaller than the width of a human hair, and they move faster than anything you'll ever touch. But here's what we didn't understand until recently. These motors are ancient, older than animals, older than plants, older than anything with tissues or organs or a body plan. They were life's first solution to an impossible problem. How do you move when you don't have muscles? How do you swim through water that feels like concrete? How do you coordinate thousands of moving parts when you don't have a brain to control them? Life solved these problems 3 billion years ago, and the solution, molecular motors spinning inside cells, became the blueprint for every movement in every living thing that would ever exist. Tonight, we're going inside the machinery of motion itself, from the first spinning engines in ancient bacteria to the coordinated arrays powering cells in your body right now. We'll explore how life learned to move in a world where the physics are alien, where swimming shouldn't work, and where motion requires solutions stranger than anything humans have engineered. Because understanding these motors changes everything. It reveals that movement didn't begin with muscles, it began with chemistry that learned to spin. This is Omni, stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. Picture a single bacterium in a drop of pond water. It's smaller than a dust mote, smaller than a pollen grain. So small that if you lined up 10,000 of them end to end, they'd barely cross the period at the end of this sentence. From your perspective, it's invisible. But from its perspective, it's navigating a universe with completely different rules. The bacterium has a tail, a flagellum, and it's spinning. Not waving back and forth like a fishtail or undulating like an eel. Spinning, like a propeller. And at the base of that tail, anchored in the cell membrane, is a motor, a molecular motor. It's built from about 30 different proteins, each one locking into place with atomic precision. There are rings that rotate, a rotor that spins, a stator that holds everything in place. It looks engineered, but no one designed it. It assembled itself, protein by protein, guided by nothing more than chemical attraction and the shape of molecules folding in just the right way. And this motor is fast. The flagellum spins at about 100,000 rotations per minute. If you scaled that up to the size of a car wheel, it would be spinning faster than a jet turbine, and it runs constantly. For hours, for days, for the entire life of the cell. It doesn't overheat, it doesn't wear out, it just keeps spinning. But here's what makes it extraordinary. It's spinning through water that doesn't behave like the water you know. At the bacterium scale, water isn't a fluid, it's thick, dense, almost solid. If you were shrunk down to bacterial size and tried to swim through water, it would feel like swimming through honey, or tar, or wet concrete. Every stroke would require enormous effort, and the moment you stopped pushing, you'd stop moving. Instantly. There's no coasting at this scale, no gliding, no momentum carrying you forward. This is the world of low Reynolds number. It's a regime of physics most of us never experience. In our world, at human scales, inertia dominates. When you throw a ball, it sails through the air. When you dive into a pool, you glide forward even after you stop kicking. Momentum carries you, but shrinks small enough, and inertia disappears. Viscosity takes over completely, and motion becomes something else entirely. Physicist Edward Purcell described it in a famous lecture in 1977. He said that at low Reynolds number, moving through water is like trying to swim in molasses while someone pours more molasses on you from above, and he proved mathematically that certain types of swimming simply can't work. If you open and close your body symmetrically, like a scallop opening and closing its shell, you go nowhere. The water you push forward on the opening stroke comes right back on the closing stroke. Zero net displacement. It's called the scallop theorem. At low Reynolds number, reciprocal motion is useless. So bacteria can't swim like fish. They can't undulate their tails back and forth symmetrically. That would leave them motionless, trapped in the viscous world. They had to invent something else. They invented rotation. The bacterial flagellum doesn't wave. It spins in one direction, like a corkscrew drilling through the viscosity. The motion is asymmetric. Every rotation is different from every other rotation in time, and that asymmetry generates thrust. Slowly, inefficiently by our standards, but it works. The bacterium moves forward at about 20 micrometers per second. That's slow in absolute terms. A snail would laugh. But relative to its body size, it's fast. 20 micrometers per second is about 10 body lengths per second. If you scaled that up to human size, you'd be running at 60 miles per hour, and the bacterium can change direction. When the motor spins counterclockwise, the flagellum pushes the cell forward in a straight line. But when the motor reverses and spins clockwise, the flagellum comes apart. Multiple flagella that were bundled together separate, the cell tumbles randomly, and when the motor switches back to counterclockwise, the bacterium shoots off in a new direction. This is chemotaxis. The cell isn't just swimming randomly, it's navigating. It can sense chemical gradients, concentrations of nutrients or toxins in the water, and it moves toward what it needs and away from what will kill it. Without a brain, without eyes, without any centralized control system, the bacterium is processing information, making decisions, and directing its motion using nothing but molecular machinery. This motor, the bacterial flagellum, is one of the oldest continuously running machines on Earth. It evolved more than 3 billion years ago, back when Earth's atmosphere had almost no oxygen, and the only life on the planet was microbial. And it's still here, still spinning, in bacteria in your gut, in the soil, in the ocean, in the clouds. Billions of trillions of these motors running simultaneously, all descended from that first molecular engine that learned to spin. But bacteria aren't the only ones who had to solve this problem. There's another kind of cell, larger, more complex, that face the same challenge. These are protists, single-celled organisms that are neither bacteria nor animals nor plants. They're eukaryotes, which means they have a nucleus and compartments inside their cells. And many of them move, but they don't use rotating flagella. They use something else, something that looks like hair. Cilia. If you look at a paramecium under a microscope, its entire surface is covered in thousands of tiny projections, all beating in coordinated waves. Each cilium is about 10 micrometers long, much shorter than a bacterial flagellum, and instead of spinning, it bends. It sweeps forward in a fast power stroke, then recovers backward in a slower return stroke. And because the motion is asymmetric, the forward stroke is fast and stiff, the return stroke is slow and flexible. It generates thrust. But here's what's remarkable. A single cilium doesn't do much. The power stroke pushes water one way, the return stroke pulls it back. At low Reynolds number, that might seem like it would cancel out, but the timing matters. The asymmetry in speed and shape means that slightly more water is pushed forward than pulled back. The net effect is small, but it's not zero. And a paramecium doesn't have just one cilium. It has about 3,000 of them, and they all beat together in perfect coordination, creating waves that ripple across the cell surface from front to back. It's called metachronal rhythm, like wind moving through a field of wheat, or fans doing the wave in a stadium. Each cilium is slightly out of phase with its neighbours, so the waves flow smoothly across the surface, generating continuous thrust. The paramecium doesn't have a brain coordinating this. It doesn't have nerves sending signals to each cilium, telling it when to beat. The coordination is mechanical. Each cilium is connected to its neighbours through the cell's internal structure, through a network of proteins and microtubules beneath the surface. When one cilium bends, it creates a slight mechanical tension that pulls on the next cilium, triggering its power stroke a fraction of a second later. The wave propagates through mechanical coupling, not neural commands. It's self-organized, emergent. The cell doesn't control the rhythm. The rhythm arises from the structure itself. And this isn't unique to paramecium. Cilia are everywhere. In your lungs, cilia beat constantly, sweeping mucus and trapped particles up and out of your airways. In your brain, cilia line the ventricles, moving cerebrospinal fluid. In the fallopian tubes, cilia move the egg toward the uterus. In your nose, in your throat, in your reproductive system, cilia are beating, moving fluids, clearing debris, transporting cells. Every cilium in your body is a descendant of the first cilia that evolved in single-celled organisms over 1 billion years ago. The structure is almost identical. At the core of every cilium is a scaffold called the axoneme, 9 pairs of microtubules arranged in a circle around a central pair. This 9+2 structure is universal. It's in paramecium, in algae, in the cells lining your lungs. And attached to those microtubules are motor proteins called dynein. Dynein is the engine. It's a molecular motor that walks along the microtubules, pulling them past each other. When dynein on one side of the cilium pulls, the microtubules slide, the cilium bends in that direction. When dynein on the other side pulls, the cilium bends back. The coordinated action of thousands of dynein motors, all pulling in sequence, creates the cilium's beat. And dynein, like the bacterial flagella motor, is powered by chemistry, specifically by a molecule called ATP, adenosine triphosphate. ATP is the energy currency of all cells. When a molecule of ATP is broken down into ADP and a phosphate group, energy is released. Dynein captures that energy and uses it to change shape, to take a step to pull on the microtubules. Each step is tiny, about 8 nanometers. But dynein takes millions of steps, and thousands of dynein motors work together, and the result is a cilium that beats 10 to 40 times per second, for hours, for days, for your entire life. This is the machinery of motion. Not muscles contracting, not neurons firing, just proteins, folded into shapes that can harvest energy from broken chemical bonds and convert it into mechanical work. These motors are so small you need an electron microscope to see them. But they're powerful enough to move cells through water, to clear mucus from your lungs, to circulate fluid through your brain. And they're ancient. Bacterial flagella evolved over 3 billion years ago. Eukaryotic cilia and flagella evolved at least 1.5 billion years ago, maybe earlier. For most of Earth's history, these molecular motors were the only way anything moved. Long before fins or legs or wings, long before muscles or bones or nervous systems, life was moving, navigating, hunting, escaping, using nothing but spinning and beating at the molecular scale. So tonight we're going deeper. We'll explore how these motors work, how they evolved, how they coordinate without commands. We'll see what happens when you try to move through a universe where viscosity dominates and inertia disappears. We'll trace the lineage from the first bacterial motor to the cilia in your lungs, revealing a continuity of motion that spans billions of years. Because motion didn't start with muscles, it started with molecules that learned to spin and beat. And every moving thing alive today carries that legacy inside. Let's begin where motion begins, with forces so small they're measured in piconewtons, and structures so precise they're built one atom at a time. Maybe motion was always going to be a problem. If you're alive, you need to find things. Food, light, safety, mates. And if you're rooted in place, you're at the mercy of whatever drifts past. You can't hunt, you can't escape. You're waiting for the world to come to you. And sometimes it doesn't. So life needed to move. But at the smallest scales, movement isn't intuitive. It's not like walking or swimming in our world, where you push off the ground or stroke through water and momentum carries you forward. At microscopic scales, the physics are fundamentally different. The rules change, and life had to figure them out from scratch. Let's start with the problem, the Reynolds number. It's a dimensionless number that compares inertial forces to viscous forces. When Reynolds number is high, like when you swim in a pool, or a bird flies through air, or a fish moves through water, inertia dominates. You can glide, you can coast. Your momentum carries you even after you stop exerting force. But when Reynolds number is low, viscosity dominates, and below a Reynolds number of about 1, the physics invert completely. A bacterium swimming through water operates at a Reynolds number around 0.01. That's 5 orders of magnitude below the threshold where inertia matters. For the bacterium, water isn't a fluid you can push against and glide through. It's a viscous medium that resists every movement and stops you the instant you stop pushing. According to research by Howard Berg at Harvard University in the 1970s and 80s, if a bacterium stops spinning its flagellum, it comes to a complete halt within a microsecond. Not a gradual slowdown, an instant stop. There's no drifting, no momentum. Motion and force are directly coupled. You move only as long as you push. Purcell explained this with a thought experiment in his 1977 lecture, Life at Low Reynolds Number. Imagine you're a bacterium, you have a little paddle, and you decide to row. You push the paddle forward through the water, then pull it back. At high Reynolds number, that works. The forward stroke moves you forward more than the backward stroke moves you back because of inertia and the way you can orient the paddle. But at low Reynolds number, there's no inertia to help you. The water is so viscous that every movement is reversible. If you push forward and pull back in the same path, you end up exactly where you started. Zero net displacement. Purcell called this the scallop theorem. A scallop opens its shell slowly, then snaps it shut quickly, and the asymmetry in speed creates thrust at high Reynolds number. But at low Reynolds number, speed doesn't matter. Only geometry matters. If your motion is reciprocal, if it looks the same played forward or backward in time, you go nowhere. So how do you move when symmetry doesn't work and momentum doesn't exist? You need asymmetry, not in speed, but in shape, in the path your motion traces through space. The bacterial flagellum solves this with rotation. The flagellum is a helical filament shaped like a corkscrew, and it spins. As it rotates, it traces a three-dimensional spiral path through the water. That spiral is asymmetric. Every point on the helix follows a different trajectory, and that asymmetry generates thrust. The bacterium is pushed forward, slowly, steadily, as long as the motor keeps spinning. But here's the elegant part. The motor itself is reversible. It can spin clockwise or counterclockwise. When it spins counterclockwise, the flagellum bundles together with other flagella on the cell, and they form a coordinated propeller that pushes the cell forward in a smooth run. But when the motor switches to clockwise, the bundle flies apart. Each flagellum goes its own way. The cell tumbles, rotating randomly in space. Then the motor switches back to counterclockwise. The flagella rebundle, and the cell shoots off in a new direction. This run and tumble behavior is how bacteria navigate. They can't sense gradients directly. They're too small to detect a difference in concentration between their front end and their back end. Instead, they measure concentration over time. As they swim, they're constantly sampling the chemical environment. If the concentration of a nutrient is increasing, if things are getting better, they run for longer before tumbling. If the concentration is decreasing, if they're swimming the wrong way, they tumble more frequently, reorienting randomly until they happen to swim in a better direction. It's a biased random walk, and it works. Studies by Berg and others in the 1980s showed that E. coli bacteria can climb chemical gradients with remarkable precision, finding food sources in complex environments using nothing but this motor and a handful of sensory proteins embedded in their membrane. The motor itself is a marvel of molecular engineering. At its core is a rotor, a set of proteins called flig that form a ring in the cell membrane. Surrounding the rotor is a stator, proteins called MOTA and MOTB that anchor to the cell wall and don't rotate. The stator proteins are proton channels. Protons, hydrogen ions, flow through them from outside the cell to inside, driven by a concentration gradient, and as each proton passes through, it pushes on the rotor, causing it to turn slightly. It's like a water wheel powered by a stream of ions. Thousands of protons flow through every second, and each one contributes a tiny push. The cumulative effect is continuous rotation at about 100,000 RPM. The efficiency is staggering. According to research by Yoshiyuki Soa and colleagues published in Nature in 2005, the bacterial flagella motor converts nearly 100% of the proton motive force into mechanical work. There's almost no energy lost as heat. For comparison, human-engineered electric motors typically operate at 70 to 90% efficiency. The bacterial motor is more efficient than anything we've built, and it self-assembles. No one puts it together. The proteins are synthesized in the cell, they diffuse to the membrane, and they find each other. They lock into place based on their shapes, complementary surfaces that fit together like puzzle pieces. The rotor forms first, then the stator proteins arrive and anchor around it. The hook and filament grow afterward, protein by protein, threaded through a hollow channel in the centre of the structure. It takes about 20 minutes for a complete flagellum to assemble, and once it's built, it starts spinning. There's no instruction manual, no blueprint external to the system. The information for how to build the motor is encoded in the shapes of the proteins themselves, molecular self-assembly, and it works every time in billions of bacteria across thousands of species. But bacteria aren't the only ones who mastered low Reynolds number motion. Eukaryotic cells, larger, more complex cells with nuclei and internal compartments, face the same problem, and they came up with a different solution. Cilia. Where bacterial flagella spin, cilia beat. They bend back and forth in an asymmetric stroke, a fast, straight power stroke in one direction and a slow, curved recovery stroke in the other. The asymmetry means they push more water forward than they pull back, generating net thrust. But a single cilium doesn't generate much force. The real power comes from having thousands of them, all beating together in coordinated waves. A paramecium has about 3,000 cilia covering its surface. Each one is about 10 micrometers long, and they're arranged in neat rows like the bristles on a brush. And they don't all beat at the same time. They beat in sequence, one after another, creating a wave that travels across the cell surface from front to back. It looks like wind rippling through a field of grain. Scientists call this metachronal rhythm, meta meaning after, and chronal meaning time. Each cilium beats just slightly after the one in front of it, and the phase delay creates the wave. But how do they coordinate? The paramecium doesn't have a brain. It doesn't have neurons sending signals to each cilium orchestrating the beat. The coordination is mechanical, built into the structure of the cell itself. At the base of every cilium is a basal body, a cylindrical structure made of microtubules that anchors the cilium to the cell surface. And beneath the surface, the basal bodies are connected to each other by a network of protein fibers called striated rootlets. These rootlets create mechanical tension. When one cilium beats, it pulls on its basal body, which pulls on the rootlets, which tug on the neighbouring basal body, which triggers the next cilium to beat. The wave propagates through direct mechanical coupling. No commands, no signals, just structure and physics. The cell doesn't tell the cilia when to beat. The cilia tell each other. And the beating is powered by dynein. Inside every cilium is a scaffold called the axoneme. 9 pairs of microtubules arranged in a circle with 2 single microtubules in the centre. This 9+2 structure is ancient and universal. It's the same in algae, in paramecia, in the cells lining your lungs. Attached to the outer microtubule pairs are motor proteins, dynein arms, that reach out and grab the neighbouring microtubule pair. When dynein binds to its target and breaks down ATP, it pulls, sliding the microtubules past each other. But the microtubules are anchored at the base, so instead of sliding freely, they bend. The cilium bows to one side, then dynein on the opposite side pulls, and the cilium bows back. The result is a whip-like beat. Research by Jonathan Howard at Yale University and others in the 1990s showed that the dynein motors are mechanically regulated. When the cilium bends too far in one direction, the microtubules on that side are stretched, which deactivates the dynein motors. Meanwhile, the microtubules on the opposite side are compressed, which activates their dynein. The system has built-in feedback. The motors know when to stop pulling and when to start pulling again, based purely on the mechanical strain in the structure. There's no external control. The cilium regulates itself, and this isn't a new invention. Cilia evolved at least 1.5 billion years ago, possibly earlier. The last common ancestor of all eukaryotes, the cell that gave rise to animals, plants, fungi and protists, almost certainly had cilia. We know this because cilia are everywhere. Unicellular algae use them to swim. Coral larvae use them to drift through the ocean. Your cells use them to move fluids and sense the environment. The structure is so conserved, so fundamental, that losing it is usually lethal. According to research published by David Mitchell at SUNY Upstate Medical University in the 2000s, mutations that disrupt the 9+2 axoneme structure cause severe diseases in humans: primary ciliary dyskinesia, polycystic kidney disease, retinal degeneration. Cilia aren't optional, they're essential. and they've been essential for over 1 billion years. So here's what we're seeing. 2 different lineages, bacteria and eukaryotes, both facing the same problem. How to move at low Reynolds number, where the rules of motion are alien and intuition from our world doesn't apply. And both lineages solved it with molecular motors. One lineage built rotary engines, the other built beating arrays. But both solutions rely on the same fundamental principles: asymmetry, continuous energy input, and mechanical coordination without central control. These motors are the foundation. Everything that moves, everything that swims, crawls, flows, or flies, descends from cells that first learned to move with engines built from folded proteins. And those engines are still running, still spinning and beating inside you right now. Let's go deeper into the machinery itself, the motors. Because when we talk about molecular motors, we're not speaking metaphorically. These aren't just proteins that happen to move. They're engines in the most literal sense, structures that convert chemical energy into mechanical work, with rotors, levers, power strokes and recovery phases. They have moving parts, they generate force, they do work against resistance, and they operate with a precision that would be impossible to achieve with anything built by human hands at that scale. The motor protein that powers cilia, dynein, is enormous by molecular standards. It's made of multiple subunits, with a combined molecular weight of about 1.5 million daltons. For context, a typical protein might be 50,000 daltons. Dynein is as giant, and its structure reflects its function. At one end of the dynein molecule is a ring of 6 protein domains arranged in a hexagon. This ring binds and breaks down ATP, the fuel molecule. At the other end is a stalk that reaches out and grabs onto a microtubule, the track it walks along. And connecting them is a linker domain that changes shape when ATP is broken down, converting chemical energy into a mechanical swing. Here's how it works. Dynein starts attached to one microtubule. Its stalk reaches out and binds to a second microtubule nearby. At this point, dynein is locked in place, holding the 2 microtubules together. Then an ATP molecule arrives. It binds to the ring. Dynein breaks the ATP into ADP and a phosphate group, and the energy released causes the linker domain to swing. The motion is like a lever pulling. The linker bends, dragging one microtubule relative to the other. The step size is about 8 nanometers. That's roughly the distance between tubulin subunits, the building blocks of microtubules. One step, one building block. Then dynein releases, resets, and takes another step, and another, and another. At full speed, a single dynein motor can take about 8 steps per second. That doesn't sound fast. But remember, there are hundreds of dynein motors attached along the length of a single cilium. They all step together, and the cumulative effect is that the microtubules slide past each other at speeds of several micrometers per second. For a structure only 10 micrometers long, that's fast. The cilium bends, sweeps forward, recovers and beats again, 10 to 40 times per second, depending on the cell type. Research by Takahide Kon and colleagues at Osaka University in 2012 used cryo-electron microscopy to visualize dynein in different states, bound, unbound, with and without ATP, and revealed the structural changes in atomic detail. The linker swings like a hinge. The stalk binds and releases in a coordinated cycle. Every part of the motor is optimized for its task. There's no wasted motion, no unnecessary complexity, just a machine that does one thing: convert ATP into sliding motion, and does it with near-perfect reliability. But dynein isn't the only motor protein. There's another one closely related called kinesin. Kinesin walks along microtubules too, but it walks in the opposite direction. Dynein generally moves toward the minus end of microtubules, toward the base of the cilium. Kinesin moves toward the plus end, toward the tip, and kinesin is faster. It can take about 100 steps per second, covering a micrometer in about 12 milliseconds. That's roughly the speed of a tortoise, if you scale it up to human size. Not fast by our standards, but at the molecular scale, kinesin is sprinting. And kinesin doesn't just walk, it carries cargo. Inside your cells, kinesin motors are constantly transporting vesicles, organelles, and protein complexes from one part of the cell to another. They're like delivery trucks on a highway system made of microtubules. The cell manufactures something in the nucleus or the endoplasmic reticulum, packages it in a membrane-bound vesicle, attaches a kinesin motor to it, and the motor walks it down a microtubule to wherever it needs to go. Without kinesin, cells couldn't move materials over long distances. Neurons, for example, are enormous. Some human neurons extend over 1 meter in length. Getting proteins from the cell body to the tip of the axon would take weeks by diffusion alone. Kinesin makes the journey in hours. Kinesin structure is simpler than dynin's. It has 2 motor domains, 2 heads, that take turns stepping forward along the microtubule, like feet walking. One head binds to the microtubule, breaks down ATP, and swings forward. The motion pulls the second head forward. That head then binds, breaks down its own ATP, and swings, pulling the first head forward again. The 2 heads alternate, and the motor walks hand over hand, or head over head, down the microtubule track. The walking motion was directly visualized in the late 1990s by Ron Vale and colleagues at UC San Francisco, using fluorescence microscopy and single molecule tracking. They attached fluorescent tags to individual kinesin molecules and watched them walk along microtubules in real time. The steps were consistent, 8 nanometers per step, perfectly matching the spacing of tubulin subunits. Kinesin doesn't slip. It doesn't stumble. It places each foot exactly where the next binding site is waiting. The precision is absolute. And here's what's remarkable about both dynin and kinesin. They operate stochastically. At the molecular scale, thermal motion is huge. Molecules are constantly being jostled by random collisions with water molecules, Brownian motion. The forces involved are on the order of piconewtons, and the energies are comparable to the thermal energy at room temperature. That means molecular motors are constantly being knocked around, buffeted by noise. Yet they still walk in one direction. They still generate force. They still transport cargo reliably. How? The answer is that the motors are designed to bias the random motion. They don't fight against Brownian motion, they harness it. When dynin or kinesin breaks down ATP, the energy release doesn't just push the motor forward, it changes the motor's shape in a way that makes it more likely to bind to the next site on the microtubule rather than the previous one. The motor is essentially lowering the energy barrier for moving forward and raising it for moving backward. It's using chemical energy to create a directional bias in an otherwise random walk. This concept, Brownian ratchets, as they're sometimes called, was explored theoretically by physicists in the 1990s and 2000s. The motor doesn't need to overpower thermal noise, it just needs to tip the statistical odds in favour of forward motion. And because ATP hydrolysis is irreversible, you can't spontaneously reassemble ATP from ADP and phosphate under cellular conditions. The motor's steps are irreversible too. The directionality is built into the thermodynamics, so every motor protein is, in a sense, extracting order from chaos, taking the random thermal jostling of molecules and converting it into directed, purposeful motion. Now scale this up. Imagine not one motor, but thousands, all working together. That's what you have in a single cilium. Hundreds of dynin motors, distributed along 9 pairs of microtubules, all pulling in coordinated bursts. The coordination doesn't come from external signals, it comes from mechanical feedback. When one set of dynin motors pulls and bends the cilium to one side, the microtubules on that side are stretched. The stretching changes the binding affinity of the dynin motors. They become less likely to stay attached. Meanwhile, the microtubules on the opposite side are compressed, which makes their dynin motors more likely to bind and pull. The result is a self-regulating oscillation. The cilium bends one way, then the other, over and over, driven entirely by the mechanical properties of the structure. This is self-organization. No central controller. No clock ticking in the background telling the motors when to fire. Just a collection of molecular machines, each following simple rules based on local mechanical strain, and the global behavior, a coordinated beat, emerges from their interactions. And it's not just cilia. The same principles apply to other motor-driven systems. Consider a sperm cell. A human sperm has a flagellum, not a bacterial-style rotating flagellum, but a eukaryotic one, powered by dynin, with the same 9+2 axoneme structure as cilia. But instead of beating like a cilium, the sperm flagellum undulates. It sends waves down its length, from base to tip, and those waves push against the fluid, propelling the sperm forward. The flagellum is about 50 micrometers long, 5 times longer than a typical coelium, and the wave propagates at a speed of about 20 micrometers per second, faster than the sperm itself swims. The wave travels down the flagellum, and as it goes, it generates thrust. The sperm moves forward at about 200 micrometers per second, about 4 body lengths per second. Not as fast as a bacterium relative to size, but fast enough. Sperm have to cover a distance of about 15 centimeters to travel from the vagina to the fallopian tube. At 200 micrometers per second, that takes about 12 minutes, assuming they swim in a straight line. In reality, it takes longer. Sperm don't swim straight. They spiral, they get stuck in mucus, they take wrong turns. But the motors keep running. The flagellum keeps beating. And eventually, if everything goes right, one sperm reaches the egg. The energy cost is enormous. Sperm are packed with mitochondria, cellular power plants that produce ATP. The mitochondria are wrapped around the base of the flagellum, forming a tight spiral called the midpiece. They produce ATP continuously, feeding it to the dynein motors, which burn through it at an astonishing rate. A single sperm uses about 10 to the 9 molecules of ATP per second. That's a billion ATP molecules every second just to keep the flagellum beating, and the sperm only has enough energy stored to swim for a few hours at most. It's a sprinter, not a marathoner. Use it or lose it, but it works. Sperm have been using this design for hundreds of millions of years. Every vertebrate, every insect, every organism with motile gametes uses some version of this system. Dynein-powered flagella, undulating or beating, all descended from the same ancient motor proteins that evolved over 1 billion years ago. And it's not just animals. Plants have motor proteins too, though they don't use them for swimming. Most plant cells are immobile, but inside plant cells, organelles are constantly being moved around by myosin motors walking along actin filaments. Chloroplasts rearrange themselves to maximize light capture. The cytoplasm streams in circular currents, distributing nutrients, and during cell division, motor proteins pull chromosomes apart, ensuring each daughter cell gets a complete set of genes. Myosin is another ancient motor protein, related to dynein and kinesin, but specialized for a different track, actin filaments instead of microtubules. Myosin is also the motor that powers muscle contraction in animals, but long before muscles existed, myosin was moving things inside single cells. The motor came first, the muscle came later. In fact, all 3 major families of motor proteins, dynein, kinesin, and myosin, appear to share a common evolutionary ancestor. They have similar structural motifs, particularly in the ATP binding domain. The details differ, but the core mechanism is the same. Bind ATP, break it down, use the energy to change shape, generate force, reset, repeat. It's a solution that evolution discovered early and refined over billions of years. And here's the stunning part. These motors are reversible in principle, but unidirectional in practice. If you could somehow push a dynein motor backward and force it to synthesize ATP from ADP and phosphate, the motor would run in reverse. But under normal cellular conditions, that doesn't happen. The concentration of ATP is high, the concentration of ADP and phosphate is lower, and the thermodynamic gradient drives the reaction in one direction. The motor walks forward, the cilium beats, the cell moves. This is life at the molecular scale. Chemistry becomes motion, energy gradients become direction, and billions of years of evolution have shaped these motors into machines so efficient, so precise, and so reliable that we're still trying to figure out how to replicate them with human engineering. But we're not done yet, because understanding how a single motor works doesn't explain how thousands of motors coordinate across an entire cell, or how cells with different motor architectures navigate toward food, away from danger, toward light, or toward each other. That's where the story gets even stranger. Because motion without a brain isn't just about motors. It's about sensing, responding, and making decisions with nothing but chemistry and structure. Moving is one thing. Moving towards something is another. If you're a single cell in a vast ocean spinning or beating your way through water, you're not just trying to move. You're trying to go somewhere useful, toward food, toward light, away from toxins, toward other cells of your kind. But you don't have eyes, you don't have a nose, you don't have a brain to process sensory information and plan a route. You have molecules embedded in your membrane, and you have motors that spin or beat. Somehow that has to be enough. And it is. Bacteria navigate chemical gradients with a precision that rivals anything more complex organisms can do. The process is called chemotaxis, and it's one of the most elegant examples of molecular computation in biology. Here's the setup. A bacterium swimming through water is surrounded by chemicals, nutrients like sugars and amino acids, toxins like acids or heavy metals. These chemicals diffuse through the water, creating gradients, regions where the concentration is higher or lower. If the bacterium could measure the concentration at two different points in space and compare them, it could figure out which direction the gradient is pointing and swim that way. But the bacterium is too small. It's only about 2 micrometers long. The difference in concentration between its front end and its back end is negligible. Lost in the noise of random molecular fluctuations, it can't sense a spatial gradient directly. So instead, it measures concentration over time. As the bacterium swims, receptor proteins on its surface are constantly binding to molecules in the surrounding water. These receptors, called chemoreceptors, change shape when they bind to their target molecule. That shape change triggers a cascade of chemical reactions inside the cell, altering the behavior of the flagellar motor. If the concentration is increasing, if the bacterium is swimming up the gradient toward higher concentrations of food, the motor keeps spinning counterclockwise, and the cell continues running straight. But if the concentration is decreasing, if the bacterium is swimming down the gradient away from food, the motor switches to clockwise, the flagella fly apart and the cell tumbles, reorienting randomly in space. Then the motor switches back and the bacterium swims off in a new direction. The result is a biased random walk. When swimming in the right direction, runs along. When swimming in the wrong direction, tumbles are frequent. Over time, the bacterium drifts up the gradient, not because it knows where it's going, but because the statistics favour it. But here's the clever part. The chemoreceptors adapt. If the bacterium swims into a region of higher concentration and stays there, the receptors gradually become less sensitive. They reset their baseline. That way, the bacterium doesn't just respond to absolute concentration, it responds to change. It's measuring the time derivative of concentration. It's asking, am I getting closer to food or farther away? Not, how much food is here right now? This adaptation happens through a process called methylation. There are enzymes inside the cell that add or remove methyl groups from the chemoreceptors, changing their sensitivity. When the receptor binds to a nutrient, methylation increases, making the receptor less responsive. When the nutrient unbinds, demethylation decreases, making it more responsive again. The system finds a balance, a steady state where the receptor is tuned to detect changes rather than absolute levels. Research by Howard Berg and Dennis Bray in the 1980s and 90s showed that E. coli bacteria can detect changes in chemical concentration as small as 0.1% over distances of a few micrometers. That's astounding sensitivity. They're measuring tiny differences in the arrival rate of molecules, filtering out noise, and using that information to control a motor. And it all happens with proteins. No neurons, no synapses, just receptors, kinases, and flagella motors interacting through chemical signals. And bacteria don't just sense chemicals. Some sense magnetic fields. Magnetotactic bacteria, discovered by Richard Blakemore in the 1970s, contain chains of magnetite crystals inside their cells. These crystals are magnetic. They align with Earth's magnetic field, and the entire chain acts like a compass needle, orienting the bacterium north or south. In the northern hemisphere, magnetotactic bacteria swim north and downward, following the magnetic field lines toward the sediment at the bottom of ponds and lakes, where oxygen is scarce, which is where they thrive. In the southern hemisphere, they swim south and downward. Same strategy, different hemisphere. The magnetite crystals are called magnetosomes, and they're synthesized by the bacteria, grown inside membrane-bound compartments with exquisite control over crystal size and shape. Each magnetosome is about 50 nanometers across, small enough to be a single magnetic domain, meaning the entire crystal acts as one tiny magnet. String a dozen of them together in a line, and you have a biological compass sensitive enough to detect Earth's weak magnetic field, which is only about 50 microteslas. But magnetotaxis isn't just about following a compass, it's about efficiency. Magnetotactic bacteria still use chemotaxis, they still sense oxygen gradients. But instead of swimming in 3 dimensions, randomly exploring all directions, they're constrained to swim along one axis, north-south. That reduces the search space. They're essentially navigating in 1 dimension instead of 3, which makes finding the optimal oxygen concentration much faster. The magnetic field isn't the destination. It's a guide rail keeping them on course. Other organisms sense light. Phototaxis, movement toward or away from light, is widespread among single-celled organisms. Algae do it, protists do it, even some bacteria do it. And the reason is simple. If you photosynthesize, light is food. You need to position yourself where the light is strong enough to power your metabolism, but not so strong that it damages your cells. You need to find the sweet spot. But sensing light at the single-cell level is tricky. You don't have a lens, you don't have a retina, you don't have image-forming optics. You need something simpler. Many algae solve this with an eye spot, a patch of pigmented proteins near one side of the cell. The pigment absorbs light, creating a shadow on the opposite side. And near the base of the flagellum there's a light-sensitive protein, a photoreceptor, that detects the intensity of light hitting the cell. As the cell rotates while swimming, the eye spot periodically blocks the light from reaching the photoreceptor. The photoreceptor experiences a flicker, bright, dim, and the frequency of the flicker depends on the cell's orientation relative to the light source. If the cell is swimming directly toward the light, the eye spot never blocks it. The photoreceptor sees constant illumination, and the flagellum beats normally. But if the cell is swimming at an angle, the eye spot passes in front of the photoreceptor once per rotation, and the flicker tells the cell it's off course. The photoreceptor triggers a response, usually a change in the beating pattern of the flagellum, that causes the cell to turn, reorienting toward the light. This system was worked out in detail in the green alga Chlamydomonas by researchers in the 1990s and 2000s. Chlamydomonas has 2 flagella, and they beat slightly differently. One is primarily responsible for forward propulsion, the other for steering. When the eye spot detects that the cell is veering away from the light source, the steering flagellum changes its beat, causing the cell to turn back. It's a feedback loop, continuously active, constantly correcting. The cell spirals toward the light, tracing a helical path through the water, homing in on the brightest region. And the photoreceptor itself, channel rhodopsin, is a light-gated ion channel. When light hits it, it opens, allowing ions to flow into the cell. That ion current changes the membrane voltage, which affects the calcium concentration near the flagella, which modulates the dynein motors. Light becomes electrical signal, becomes chemical signal, becomes mechanical motion, all in milliseconds. Channel rhodopsin, by the way, is the same protein that neuroscientists now use in optogenetics, using light to control neurons in the brains of mice and other animals. The technique was borrowed directly from algae. Evolution invented it first, for navigation. We adapted it for research. But not all sensing is about following gradients. Sometimes it's about responding to touch. Mechanotransduction, the conversion of mechanical force into biochemical signals, is fundamental to how cells interact with their environment. When a cilium bends because it hits an obstacle, or when a cell is compressed by neighbouring cells, or when fluid flow creates shear stress on a membrane, the cell needs to know. And it does, through mechanosensitive proteins embedded in its membrane. These proteins are essentially molecular springs. When the membrane is stretched or compressed, the protein changes shape, opening a channel that allows ions to flow through. The ion flux changes the cell's internal chemistry, triggering responses. Maybe the cell stops moving, maybe it changes direction, maybe it reinforces its membrane, maybe it divides. One of the best studied mechanosensors is the MSCL channel in bacteria, mechanosensitive channel of large conductance. It's a pore in the membrane that opens when the membrane is stretched. If a bacterium suddenly finds itself in a low-salt environment, say, fresh water, after living in seawater, water rushes into the cell by osmosis, and the membrane swells. Without a release valve, the cell would burst. But Mesquiel opens, releasing ions and small molecules, reducing the internal pressure and preventing lysis. It's an emergency relief valve, activated purely by mechanical tension. In eukaryotic cells, mechanotransduction is even more sophisticated. The cilia on your cells aren't just for moving fluids, they're also sensors. When fluid flows over a cilium, the cilium bends, and that bending is detected by proteins at the base. The cell receives information about flow rate, flow direction, and shear stress. This is how kidney cells sense urine flow, how cells in blood vessels sense blood flow, how cells in your inner ear detect sound vibrations and head movements, giving you the sense of balance. And sometimes mechanotransduction coordinates collective behavior. Take Volvox, a multicellular green alga that forms a hollow sphere about 0.5 millimeter across. Each cell in the sphere has 2 flagella, and they all beat in coordination, spinning the sphere as it swims through the water. But the cells on one side of the sphere beat slightly faster than the cells on the other side, creating a front and a back, an axis of polarity. The faster beating cells are pulled more taut by the surrounding extracellular matrix, and that mechanical strain feeds back into their beating pattern, reinforcing the asymmetry. The colony knows which way is forward, not because there's a command center, but because the geometry and mechanics of the structure create a stable pattern. Mechanical feedback, chemical signals, electrical gradients, all of this is happening simultaneously in every swimming cell, coordinating movement without a brain. And then there's predation, because not all movement is about finding nutrients in the water. Sometimes it's about finding other cells and eating them. Didinium is a ciliate, a relative of paramecium that hunts other protists. It's barrel-shaped, covered in cilia, and it swims through the water scanning for prey. Its preferred target is paramecium. When a didinium encounters a paramecium, it doesn't just bump into it passively. It fires trichocysts, harpoon-like structures that shoot out from the cell surface, penetrating the paramecium's membrane and anchoring the 2 cells together. Then didinium opens its oral groove, a specialized region that can expand enormously, and begins to engulf the paramecium. The entire prey cell, often as large as the predator itself, is pulled inside, where it will be digested over the next several hours. The hunting behavior is complex. Didinium doesn't swim randomly. According to research by Akihiro Ishijima and others in the early 2000s, didinium responds to chemical cues released by paramecium, detection at a distance, possibly through chemoreceptors similar to those in bacteria. When it detects prey, its swimming pattern changes. It spirals more tightly, staying in the region where the chemical signal is strongest. When contact is made, the trichocysts fire automatically, a mechanical trigger, like stepping on a landmine. This is predation at the cellular level, hunting strategies, weapons, digestion, all orchestrated by molecular motors, sensory proteins, and self-organizing membranes. No nervous system, no muscles. Just a single cell with thousands of cilia beating, trichocysts loaded, and a hunger that drives it through the microscopic world in search of prey. And paramecium, for its part, has defenses. It can swim away, fast. It can reverse the beating direction of its cilia, swimming backward to escape. Some species even have trichocysts of their own, which they discharge when attacked, creating a cloud of tangled proteins that confuses or repels predators. It's an arms race, playing out at the microscopic scale, driven by the same evolutionary pressures that shape predator-prey dynamics in the visible world. And all of it, every escape, every chase, every navigation toward light or food or safety comes down to motors, molecular machines, spinning or beating, coordinated by nothing more than chemistry and structure. No brain required, just motion, refined over billions of years into something that looks for all the world like intentionality. So where did all of this come from? Because molecular motors didn't just appear, fully formed, spinning at 100,000 RPM, or walking along microtubules with 8 nanometer precision. They evolved. They had ancestors. And somewhere, billions of years ago, there was a first motor, a protein that learned to convert chemical energy into directed motion, setting the foundation for every movement that would follow. Tracing that origin is difficult. Proteins don't fossilize. DNA degrades. The molecular record of early life is fragmentary at best. but we can make inferences. We can compare modern motor proteins across different organisms, look for shared structural features, and reconstruct what the ancestral motor might have looked like. And what we find is this: all the major motor proteins, dynein, kinesin, myosin, share a common core. They have similar ATP binding domains. They undergo similar conformational changes when ATP is hydrolyzed. They generate force through similar mechanical principles. The details differ. Dynein is huge and complex. Kinesin is smaller and simpler. Myosin is specialized for actin instead of microtubules, but the underlying mechanism is the same. This suggests a common evolutionary origin. Not that one motor evolved from another, but that all of them evolved from some ancestral ATPase, an enzyme that could bind and break down ATP long before it was harnessed for mechanical work. ATPases are everywhere in biology. They're enzymes that hydrolyze ATP, releasing energy that drives chemical reactions. Pumps that move ions across membranes are ATPases. Chaperones that fold proteins are ATPases. Helicases that unwind DNA are ATPases. The ability to bind ATP and extract energy from it is ancient, fundamental, and universal. At some point, one of these ATPases acquired the ability to change shape in a way that generated force against a substrate. Maybe it was bound to a filament, an early version of actin or tubulin, and when it broke down ATP, the conformational change pushed or pulled on the filament. That's not yet a motor in the full sense. It's just a protein that twitches when it consumes energy. But that twitch, repeated over and over, could move things, slightly, incrementally. And if that movement gave the cell even a tiny advantage, better access to nutrients, faster escape from toxins, natural selection would amplify it. Over millions of years, the protein would be refined. Mutations that made the conformational change more efficient, more directional, more forceful would be favoured. Proteins that could bind to the filament more tightly, release more predictably, reset more quickly, would outcompete those that couldn't. And eventually you'd have a motor, a protein specialised for converting ATP into motion, with a stepping cycle, a power stroke, and the ability to walk or slide along a track. This probably happened multiple times, independently, in different lineages. Myosin evolved to work with actin filaments. Dynein and kinesin evolved to work with microtubules. The bacterial flagellar motor evolved to spin in the membrane. The solutions are different because the problems were different. But they all share the same basic logic. Harness the energy from ATP hydrolysis, convert it into a shape change, use that shape change to generate force, and repeat. Now, here's where it gets interesting. Because there's one motor protein that isn't just moving things, it's making ATP. ATP synthase. It's the enzyme that produces most of the ATP in your cells, and it's also a rotary motor, arguably the most important motor in all of biology. ATP synthase sits in the membrane of mitochondria, and in bacteria it sits in the cell membrane. It has two main parts, a rotor embedded in the membrane, and a catalytic head that sticks out into the cytoplasm. The rotor spins, driven by a flow of protons across the membrane. As it spins, the catalytic head synthesizes ATP from ADP and phosphate. The rotation is coupled directly to chemical synthesis. Spin the motor one way and you make ATP, spin it the other way and you break down ATP, and that's exactly what happens. In mitochondria, the proton gradient drives the motor forward, synthesizing ATP. But in some bacteria, when ATP is abundant and the proton gradient is weak, the motor runs in reverse. It consumes ATP and pumps protons, maintaining the gradient. ATP synthase is reversible. It's both a motor and a generator. The structure was worked out by John Walker and colleagues in the 1990s, earning Walker a Nobel Prize in 1997. The catalytic head has 3 active sites arranged in a triangle. As the rotor spins, it pushes on a central stalk that runs through the middle of the head, and that stalk forces the active sites to cycle through 3 different shapes: open, loose, and tight. In the open state, ADP and phosphate bind. In the loose state, they're held in position. In the tight state, they're squeezed together so hard that they spontaneously form ATP. Then the site opens again, releasing the ATP, and the cycle repeats. 3 active sites cycling out of phase, driven by one continuous rotation. It's mechanochemistry, mechanical force directly driving a chemical reaction, and the rotor spins fast. At full speed, ATP synthase rotates at about 150 rotations per second. That's 9,000 RPM, and every full rotation produces 3 molecules of ATP, one from each active site. So a single ATP synthase molecule can produce about 450 ATP per second. Your body has trillions of them, running continuously, generating the roughly 60 kilograms of ATP you synthesize and consume every day just to stay alive. But here's the kicker: the same structure exists in bacteria. ATP synthase is ancient. It predates mitochondria. It predates eukaryotes. It was probably present in the last universal common ancestor, the organism from which all life on Earth descends. That means ATP synthase is at least 3.5 billion years old, possibly older. And when mitochondria evolved, when an ancient bacterium was engulfed by an early eukaryotic cell and became a permanent resident, an endosymbiont, it brought its ATP synthase with it. That bacterial motor is still in your mitochondria, still spinning, still making the ATP that powers every motor protein in your body. So the motor that makes the fuel also works like the motors that consume the fuel. ATP synthase spins, dynein walks, kinesin walks, myosin pulls. The bacterial flagellum spins. They're all variations on the same theme, proteins that convert energy gradients into motion. And this raises a profound evolutionary point. Once life figured out how to build molecular motors, it could do almost anything. Motors enabled motility. Cells could swim, hunt, escape. Motors enabled transport. Cells could grow larger because they could move materials internally over long distances. Motors enabled cell division. Motor proteins pull chromosomes apart during mitosis, ensuring each daughter cell gets a complete genome. Motors enabled multicellularity. When cells started forming colonies, they needed to coordinate, moving in concert, sharing resources, differentiating into specialized types. Motors made that possible. Consider the transition from single cells to multicellular organisms. It's one of the major evolutionary transitions in the history of life, and it happened multiple times, in animals, plants, fungi, algae. But every time it happened, motor proteins were involved. Take Volvox again. It's a colony of several thousand cells, each with 2 flagella, all beating together to spin the sphere through the water. But the cells aren't just identical copies. There's differentiation. Some cells are somatic cells. They swim and photosynthesize but don't reproduce. Others are germ cells. They divide and form the next generation. The colony has polarity, a front and a back. It has coordination. Cells on one side beat faster than cells on the other. And it has development. Germ cells undergo a complex series of divisions and rearrangements to form daughter colonies inside the parent, which are then released to live independently. All of this is orchestrated by motors, the flagella beating, the cytoplasmic bridges connecting cells, the contractile mechanisms that invert the developing daughter colonies during embryogenesis. None of it would work without molecular motors, coordinating action across thousands of cells. And the same is true in animals. The cilia in your lungs sweeping mucus upward, the flagellum on a sperm cell swimming toward an egg, the contractions of your heart driven by myosin pulling on actin filaments in cardiac muscle, the neurons in your brain transporting neurotransmitter vesicles down axons using kinesin. All of it traces back to the same molecular machinery that evolved billions of years ago in single cells. But scaling up motor coordination wasn't trivial. A single cell with a few thousand, cilia can coordinate through mechanical coupling, each cilium pulling on its neighbours, waves propagating through direct physical connection. But a multicellular organism with trillions of cells, spread across centimetres or metres of space, can't rely on mechanical coupling alone. You need other systems, chemical signalling, electrical signalling, nervous systems, hormones. These are the mechanisms that let multicellular organisms coordinate motor activity on a larger scale. But even then, the motors themselves haven't changed much. The dynein in your lung cilia is structurally almost identical to the dynein in a single-celled paramecium. The myosin in your muscles is closely related to the myosin in amoeba, which use it to crawl across surfaces by extending pseudopods. Evolution didn't reinvent motors when organisms got bigger, it reused them. It placed them in new contexts, under new control systems, in new combinations. But the motors themselves, the molecular machines doing the work, are ancient, conserved, and universal. And this conservation tells us something profound. Once evolution solves a problem well, it doesn't discard the solution. It builds on it, it adapts it, it scales it. But the core machinery remains. You can see this in the structure of cilia and flagella. The 9+2 axoneme, 9 pairs of microtubules in a circle, 2 in the centre, dyne in arms reaching between them, is the same in algae, in protists, in the cells of your lungs, in sperm, in the sensory cells of your inner ear. It's been conserved for over 1 billion years. Mutations that disrupt it are almost always lethal or severely debilitating. You can't mess with a 9+2 structure without breaking motility entirely, but you can modify it. Some organisms have 9+SD cilia, no central pair. These are usually sensory cilia, not motile ones. They don't beat, they just sit there, detecting chemical or mechanical signals. The motor proteins are gone, but the scaffold remains, repurposed for a different function. Other organisms have more elaborate structures. Some ciliates have compound ciliary organelles, cirri, where multiple cilia fuse together into a leg-like structure that the cell uses to walk along surfaces. The individual cilia are still there, still beating, but they're coordinated into a higher order unit. It's like fingers forming a fist. The basic components are the same, the arrangement is new, and in animals cilia diversified even further, motile cilia for moving fluids, primary cilia for sensing, nodal cilia for establishing left-right symmetry during embryonic development. These are cilia in a structure called the node, and they rotate in a specific direction, creating a leftward flow of fluid that tells the embryo which side is which. If the nodal cilia don't beat, or if they beat in the wrong direction, you get situs inversus, organs on the wrong side of the body, your heart on the right, your liver on the left. All because molecular motors didn't spin the way they were supposed to during development. That's how fundamental these motors are. They don't just move cells, they pattern bodies, they establish axes, they determine left from right, and they do it with the same basic machinery. Proteins, ATP, microtubules, that bacteria were using 3 billion years ago to swim toward food. So when we ask, where did molecular motors come from? The answer is, they came from chemistry that learned to move, from ATPases that twitched, from twitches that became steps, from steps that became coordinated walks, spins, beats, and from those first motors, everything else followed. Motility, transport, division, coordination, multicellularity, sensation, development. Every major innovation in the history of life has been, at some level, an innovation in how to use motors, and those motors are still running, in you, in every organism, in every moving cell, an unbroken chain of motion stretching back billions of years, powered by the same molecules that were spinning and beating in the earliest oceans. Now let's bring this home, literally, to your body, because everything we've been talking about, molecular motors, ciliary beating, coordinated waves, mechanotransduction, isn't just happening in pond water or ancient oceans, it's happening inside you, right now, in your lungs, your brain, your ears, your nose, your reproductive system. The same motors that evolved billions of years ago are still running, still beating, still keeping you alive. Start with your airways. Every breath you take pulls in air and with it dust, pollen, bacteria, fungal spores, smoke particles if you're near a fire, pollution if you're in a city, viral particles if someone nearby is sick. Your lungs are constantly being bombarded with foreign material, and if that material isn't cleared, it accumulates, bacteria multiply, infections set in, your lungs fill with mucus and debris, you can't breathe, but that doesn't happen, not usually, because lining your airways, from your trachea down through your bronchi and bronchioles, are millions of ciliated cells. Each cell has about 200 cilia on its surface, and those cilia are beating continuously, 10 to 15 times per second, 24 hours a day, for your entire life. They never stop. The cilia are submerged in a thin layer of mucus, a sticky gel produced by specialized goblet cells scattered among the ciliated cells, Particles that land in your airways get trapped in the mucus, and the cilia, beating in coordinated metachronal waves, push the mucus upward, against gravity, up from the depths of your lungs, through the bronchi, up the trachea into your throat. The mucus, carrying all the trapped debris, reaches the back of your throat and you swallow it, or you cough it out. Either way, it's gone, cleared from your lungs. This process is called mucociliary clearance, and it's one of the most important defense mechanisms your body has. Without it, every respiratory infection would be life-threatening, and it depends entirely on cilia, molecular motors, beating in rhythm, coordinated through mechanical coupling, powered by dynein and ATP. The coordination is remarkable. The cilia don't all beat in phase. If they did, they'd just churn the mucus in place. Instead, they beat in waves, each cilium slightly out of phase with its neighbours, creating a travelling wave that moves from the bottom of the lungs upward. It's the same metachronal rhythm we saw in paramecium, scaled up across millions of cells, spanning the entire respiratory tree. And the beat itself is asymmetric. During the power stroke, the cilium extends fully, reaching up into the mucus layer and sweeps forward, pushing the mucus along. During the recovery stroke, the cilium bends close to the cell surface, moving back through the watery layer below the mucus, where there's less resistance. The asymmetry ensures that more mucus is pushed forward than pulled back. Net flow, upward, continuous, always clearing. But when the cilia fail, everything falls apart. Primary ciliary dyskinesia, PCD, is a genetic disorder caused by mutations in the genes that encode ciliary proteins. Most commonly, the dynein motors are defective. The cilia are still there, structurally intact, but they can't beat properly. Some beat weakly, some beat erratically, out of sync, some don't beat at all. The result is devastating. Mucus accumulates in the lungs. Bacteria thrive in the stagnant mucus, causing chronic respiratory infections. Patients develop bronchiectasis, permanent damage to the airways. Lung function declines over time. Many patients need daily chest physiotherapy to manually clear the mucus, pounding on their chest to dislodge it, because their cilia can't do the job. And it's not just the lungs. People with PCD often have chronic sinus infections, because the cilia in the nose and sinuses are also paralysed. They have chronic ear infections, because the eustachian tubes, which drain fluid from the middle ear, are lined with cilia that can't clear the fluid. Male infertility is common, because sperm can't swim without functional flagella, and about half of PCD patients have situs inversus, organs on the wrong side of the body, because the nodal cilia that establish left-right asymmetry during embryonic development didn't beat properly. One defect in one type of motor protein, and the consequences cascade across multiple organ systems. That's how fundamental these motors are. They're not optional, they're not redundant, they're essential. And PCD isn't the only ciliopathy. There are dozens of genetic diseases linked to ciliary dysfunction. Polycystic kidney disease, cysts form in the kidneys because sensory cilia on kidney cells can't detect fluid flow properly, and the cells overproliferate in response. Retinitis pigmentosa. Photoreceptor cells in the retina degenerate because the connection between the inner and outer segments of the cell, which is a modified cilium, breaks down. Bardet-Biedl syndrome, obesity, cognitive impairment, polydactyly, kidney disease, all caused by defects in proteins that traffic through cilia. Cilia are everywhere, and when they break, everything breaks. But cilia aren't just for moving things, they're also sensors. Most of your cells have a single primary cilium, a non-motile cilium that sticks out from the surface like an antenna. It doesn't beat, it just sits there, but it's packed with receptors and signaling proteins, and it detects changes in the environment, chemical signals, mechanical forces, even changes in fluid flow. In your kidneys, the epithelial cells lining the tubules have primary cilia that extend into the flowing urine. When urine flows past, it bends the cilium. That bending opens mechanosensitive channels. Calcium flows into the cell, and the cell adjusts its behaviour, regulating ion transport, controlling cell division, maintaining tissue homeostasis. If the cilia are missing or defective, the cells lose their ability to sense flow, and they start proliferating uncontrollably, forming cysts. That's polycystic kidney disease. In your blood vessels, endothelial cells, the cells lining the inside of arteries and veins, have primary cilia that detect blood flow. When blood flows smoothly, in one direction, the cilia are bent consistently and the cells remain quiescent. But in regions where flow is turbulent, near arterial branches or in areas of narrowing, the cilia are jostled irregularly and the cells respond by activating inflammatory pathways. This contributes to atherosclerosis, plaque formation in arteries, because turbulent flow, sensed by cilia, triggers the cascade of events that leads to vessel damage. Even in your brain, cilia are sensing. The ventricles, fluid-filled spaces deep in the brain, are lined with ependymal cells, and those cells have motile cilia that beat constantly, circulating cerebrospinal fluid. But they also have sensory roles. The cilia detect pressure, flow rate, and chemical composition of the CSF. If the cilia stop beating, or if the flow is obstructed, fluid accumulates, pressure increases, and you get hydrocephalus, water on the brain. It's dangerous, often fatal if untreated, and it's fundamentally a problem of ciliary function. Your sense of smell depends on cilia too. The olfactory epithelium, the patch of tissue at the top of your nasal cavity, is where you detect odours. Each olfactory sensory neuron has a single dendrite that extends to the surface of the epithelium, and at the tip of that dendrite are 10 to 30 cilia. These cilia are non-motile, but they're covered in olfactory receptors, proteins that bind to odour molecules. When you breathe in, air-carrying volatile chemicals swirls through your nasal cavity. The chemicals dissolve in the mucus layer and bind to the receptors on the cilia. The binding triggers a signal cascade inside the neuron, and within milliseconds the neuron fires an action potential that travels to your brain, where it's interpreted as a smell. The cilia massively increase the surface area available for receptors. A single neuron with 20 cilia has about 20 times more receptors than it would with just a cell body. That means 20 times more opportunities to capture odour molecules. That's why your sense of smell is so sensitive. You can detect some chemicals at concentrations as low as parts per trillion. It's molecular detection, enabled by ciliary architecture. And when those cilia are damaged, by infections, by toxins, by head trauma, you lose your sense of smell. Anosmia. It's more common than people realise. Upper respiratory infections, like the common cold, can damage the cilia temporarily, causing anosmia that lasts days or weeks. COVID-19 famously caused prolonged loss of smell in millions of people, likely by damaging the supporting cells around the olfactory neurons, disrupting the environment the cilia need to function. But it's not just smell. Your sense of taste also depends on microvilli, tiny projections on taste receptor cells that are structurally similar to cilia, though built from a different protein scaffold. Taste and smell are tightly linked, both relying on surface projections to maximize receptor density. And then there's your sense of balance and hearing. In your inner ear, there are specialized sensory cells called hair cells. Each hair cell has a bundle of stereocilia, not true cilia, but elongated microvilli, arranged in a specific pattern on its surface. The tallest ones are on one side, the shortest on the other, creating a gradient. And at the base of the tallest stereocilium is often a single true cilium, the kinocilium. When sound waves enter your ear, they vibrate the eardrum, which moves the bones of the middle ear, which push on the cochlea, a fluid-filled spiral in the inner ear. The fluid moves, bending the stereocilia on the hair cells, and when the stereocilia bend toward the tallest one, toward the kinocilium, mechanosensitive channels open. Ions flood in. The hair cell depolarizes. It releases neurotransmitter onto a sensory neuron, which fires an action potential that travels to your brain. You perceive sound. The precision is extraordinary. Hair cells can detect movements as small as a fraction of a nanometer, less than the width of an atom. They can respond in microseconds, and they can distinguish frequencies over a range of about 20 Hz to 20,000 Hz, because different hair cells along the cochlea are tuned to different frequencies based on their position and the stiffness of the surrounding structures. And it's the same for balance. In the vestibular system, the part of the inner ear that detects head movements and orientation, hair cells are embedded in gel-like structures that shift when your head moves. The stereocilia bend, channels open, signals fire, and your brain knows which way you're tilted, whether you're accelerating, whether you're spinning. That's how you can walk without falling over, how you can track moving objects with your eyes, how you maintain your sense of up and down even when your eyes are closed. All of it depends on ciliary and ciliary-like structures. Projections from cells, packed with mechanosensors, converting physical motion into electrical signals. And again, when these systems fail, the consequences are severe. Genetic mutations in the proteins that form stereocilia cause hereditary hearing loss. Exposure to loud noise damages and destroys hair cells, causing permanent hearing loss. They don't regenerate in mammals, so once they're gone, they're gone. Infections can damage the vestibular hair cells, causing chronic dizziness and balance problems. So here's the picture. Every breath you take is cleared by cilia. Every odour you smell is detected by cilia. Every sound you hear is transduced by ciliary-like structures. Your kidneys sense their environment with cilia. Your blood vessels sense flow with cilia. Your brain circulates its fluid with cilia. Your embryonic development depended on cilia to tell your cells which side is left and which is right. You're alive because of molecular motors, not metaphorically, literally. Dynein walking along microtubules, powered by ATP, beating in rhythm, coordinated through mechanical coupling and chemical feedback. The same motors that evolved in single-celled organisms billions of years ago. And you didn't build them, you inherited them. They were passed down through every generation, from the first eukaryotes to the first animals to the first vertebrates to the first mammals to you. The dynein in your lungs is the descendant of dynein in ancient algae. The structure is conserved, the function is conserved, the mechanism is conserved. Evolution doesn't start over, it tinkers, it reuses, it adapts what already works, and molecular motors worked so well, so early, that life has been using them ever since. But there's one more place we need to look, one more system where these motors are absolutely critical, where their function determines whether life continues or stops. Reproduction. Let's talk about sex, cellular sex. Because reproduction, the ability to create the next generation, depends entirely on molecular motors. In almost every sexually reproducing organism, at least one gamete has to move. Usually it's the small one, the sperm, the pollen, the motile gamete that has to travel, sometimes vast distances on a cellular scale, to find and fuse with the egg. And that journey is powered by the same motors we've been talking about. Dynein, microtubules, the 9-pol-2 axoneme, the beating flagellum. Human sperm are among the most specialised cells in your body. They're stripped down, streamlined, built for one purpose, to swim. The head contains the nucleus, tightly packed DNA, condensed to about 1/6 the volume of a normal cell nucleus. There's barely any cytoplasm, no ribosomes to speak of, no machinery for making new proteins. The sperm can't repair itself, it can't grow, it can't divide. It's a missile, launched with a finite fuel supply and a single mission. And the engine is the flagellum. It's about 50 micrometers long, roughly the length of the head. And it consists of the same 9-pol-2 axoneme structure found in every motile cilium and flagellum across eukaryotes. 9 pairs of microtubules in a ring, 2 in the centre, dynein arms reaching between them, pulling and sliding, bending the flagellum in waves that propagate from base to tip. But sperm have an additional structure that cilia don't, the fibrous sheath. It's a protein scaffold that runs along the length of the flagellum, providing structural support and elasticity. It makes the flagellum stiffer, more spring-like, so that when the dynein motors pull on one side, the flagellum doesn't just collapse, it bends, stores elastic energy, and snaps back. The result is a smooth, undulating wave rather than an erratic flail. And wrapped around the base of the flagellum, just behind the head, is the midpiece, a tightly coiled spiral of mitochondria. About 75 mitochondria, arranged end-to-end like beads on a string, forming a helix around the axoneme. These mitochondria are the power plants. They produce ATP through oxidative phosphorylation, the same process happening in the mitochondria and every other cell in your body, but here it's happening at maximum capacity. Because sperm need a lot of energy. According to research published in the early 2000s by David Clapham and colleagues at Harvard Medical School, a swimming sperm consumes about 10.9 molecules of ATP per second. That's a billion ATP molecules every second. The flagellum is beating at about 10 Hz, and every beat requires thousands of dynein motors to step, slide, and reset. The ATP is consumed as fast as it's produced. The mitochondria are running at full output constantly, and the fuel is finite. Sperm carry some glycogen, a small reserve of glucose polymer, but it doesn't last long. Once it's depleted, the sperm slow down. The flagellum beats less vigorously, eventually it stops. The sperm die. They have a lifespan measured in hours to days depending on the environment. In the female reproductive tract, with access to nutrients secreted by the surrounding tissue, they can survive for up to 5 days. Outside the body, they die much faster, minutes to hours. So the clock is ticking from the moment they're ejaculated, and they have a long way to go. During ejaculation, about 200 to 300 million sperm are deposited in the vagina. The distance from the vagina to the site of fertilization, the fallopian tube, is about 15 to 18 centimeters. That doesn't sound far, but remember, a sperm is only about 60 micrometers long. Traveling 15 centimeters is the equivalent of a human swimming 2,500 kilometers, the distance from New York to Los Angeles, across the country. And sperm don't swim in a straight line, they spiral. The flagellum beats asymmetrically, generating thrust that pushes the head forward, but also rotates it. The sperm traces a helical path through the fluid, like a corkscrew drilling through space. This is partly due to the off-center attachment of the flagellum. It doesn't emerge from the exact center of the head, so the thrust is angled, causing rotation. And this is actually advantageous. The spiraling motion stabilizes the trajectory. It prevents the sperm from tumbling randomly due to Brownian motion and fluid turbulence. But even swimming at full speed, about 200 micrometers per second, it would take hours for a sperm to swim the full distance, and most of them don't make it. The vagina is acidic, hostile to sperm. Many die immediately, others get trapped in cervical mucus, which is thick and sticky, a barrier that filters out slow or malformed sperm. Only the fastest, strongest swimmers make it through the cervix into the uterus. Once in the uterus, the environment is better, closer to neutral pH, less hostile, but the journey is far from over. The uterus is a large cavity, relatively speaking, and sperm have to navigate across it to find the opening to the fallopian tube. And there are 2 fallopian tubes, one on each side. Only one of them on average contains an egg during any given menstrual cycle. Sperm that enter the wrong tube reach a dead end. Their journey ends in failure. But the ones that find the correct tube still have work to do. The fallopian tube is lined with ciliated cells, millions of them, and their cilia are beating, creating a current that flows downward toward the uterus. The egg is swept down this current after ovulation, drifting passively toward the uterus, but the sperm are swimming against it. They're fighting a current generated by the same type of motor proteins that power their own flagella. Dynein versus dynein, cellular motors on both sides of the interaction. And this is important. The ciliary current in the fallopian tube doesn't just transport the egg, it also selects for the strongest sperm. Only sperm that can swim powerfully enough to overcome the current reach the upper part of the tube, where fertilization typically occurs. It's a filter, a physical barrier that ensures only the most motile sperm succeed. When a sperm finally encounters an egg, it doesn't just bump into it and fuse. It has to get through layers of protection. The egg is surrounded by cumulus cells, follicle cells that stick to the outside of the egg, embedded in a matrix of glycoproteins. Sperm release enzymes from the acrosome, a specialized compartment in the head, that digest through this matrix, creating a path, and the flagellum keeps beating, driving the sperm forward through the dissolving barrier. Once through the cumulus, the sperm reaches the zona pellucida, a thick glycoprotein shell around the egg itself. The sperm binds to receptors on the zona, and this triggers the acrosome reaction. The acrosome ruptures, releasing more enzymes that digest a hole in the zona. The sperm burrows through, flagellum still beating, pushing forward until finally it reaches the egg membrane, and then fusion. The membranes merge, the sperm nucleus enters the egg. The flagellum is left outside, discarded. It's done its job. The motor that brought the sperm this far is no longer needed. The egg will degrade it, breaking down the microtubules, the dynein, the mitochondria. None of it passes to the next generation. but the information does. The genes encoding dynein, tubulin, the proteins that built the flagellum, they're in the nucleus, and they'll be passed on. The next generation will build its own motors, using the same instructions, and the egg, for its part, doesn't move on its own. It has no flagellum, no motile cilia. It's passive, but it's carried. The cilia lining the fallopian tube beat constantly, moving the egg toward the uterus. If the egg is fertilized along the way, it continues drifting, dividing as it goes, until it reaches the uterus and implants. If the cilia aren't beating, if there's a ciliary defect, an infection that damages the epithelium, or scarring from surgery, the egg doesn't move. It can get stuck. If it's fertilized while stuck in the fallopian tube, you get an ectopic pregnancy, a pregnancy in the wrong place. The embryo grows in the tube, which can't support it, and eventually the tube ruptures. It's a medical emergency. And it's fundamentally a failure of ciliary transport. So both gametes, sperm and egg, depend on molecular motors. The sperm's motor moves the sperm. The egg's transport is driven by motors in the cells around it. Reproduction is impossible without them, and once fertilization happens, motors become even more critical, because the fertilized egg has to divide, over and over, growing from 1 cell to 2 to 4 to 8 to trillions. And every division requires motors. During mitosis, cell division, the chromosomes in the nucleus condense, the nuclear envelope breaks down, and the chromosomes attach to a structure called the mitotic spindle. The spindle is made of microtubules, radiating from 2 poles on opposite sides of the cell. Motor proteins, kinesin and dynein, pull on the microtubules, positioning them, adjusting their length, sliding them past each other. Each chromosome has a kinetochore, a protein complex at its centromere that attaches to microtubules from both poles. The microtubules pull in opposite directions, and the chromosome is held in tension, balanced at the center of the cell. This is metaphase. The chromosomes line up in a plane halfway between the 2 poles. Then, when everything is aligned correctly, the cell triggers anaphase. The connection holding the 2 sister chromatids together is severed, and motor proteins pull the chromatids apart. Dynein at the poles reels in the microtubules, shortening them, pulling the chromatids toward opposite ends of the cell. At the same time, kinesin walks along the microtubules, pushing the poles apart. The cell elongates, the chromatids separate. By the end of anaphase, each daughter cell has a complete set of chromosomes, and then the cell divides, cytokinesis. In animal cells, a contractile ring of actin and myosin forms around the equator of the cell and tightens, pinching the cell in 2. Myosin pulls on actin filaments, sliding them past each other, constricting the membrane like a drawstring. The cell splits. 2 daughters, each with a nucleus, each with a complete genome, each ready to divide again. Without motor proteins, none of this works. Chromosomes don't separate, cells can't divide, development stops, the organism dies. This is why mutations in motor proteins are often lethal. If dynein is completely non-functional, sperm can't swim, cilia can't beat, chromosomes can't segregate, the organism doesn't survive. Even partial loss of function, like in primary ciliary dyskinesia, causes severe disease. Motor proteins aren't just useful, they're necessary. And during embryonic development, motors do even more than move chromosomes. They move entire cells. In the early embryo, cells migrate. They move from one part of the embryo to another, guided by chemical signals crawling over and around each other, rearranging themselves into the structures that will become organs. This migration is driven by the cytoskeleton, actin filaments and microtubules, and by motor proteins walking along those filaments, pulling the cell forward. Neural crest cells, for example, arise near the developing spinal cord and then migrate long distances to form structures all over the body: facial bones, peripheral nerves, pigment cells in the skin. They crawl through the embryo using myosin and actin, extending protrusions called lamellipodia at the leading edge, anchoring to the surrounding tissue and pulling themselves forward. If the motor proteins don't work, the cells don't migrate. The structures they're supposed to form never develop. You get severe congenital defects, and we've already talked about nodal cilia in the embryonic node that rotate in a specific direction, creating a leftward flow of fluid. That flow carries signaling molecules to the left side of the embryo, activating genes that specify left side identity. If the nodal cilia don't beat, or if they beat weakly, or if they rotate the wrong direction, the signaling is disrupted. You get randomized left-right patterning. 50% of the time, organs develop normally. 50% of the time, they're reversed. Situs inversus. So motors determine body plan. They establish axes. They move cells where they need to go. They transport molecules to the right places at the right times. Development is a choreography of motion, and molecular motors are the dancers. And even after development is complete, even in an adult organism, motors are essential for maintenance. Cells in your gut lining turn over every few days. Cells in your skin are constantly being replaced. Blood cells are produced continuously in the bone marrow. Every new cell comes from division of an existing cell. And every division requires motors to pull the chromosomes apart. Your neurons, on the other hand, don't divide. Most of them are as old as you are, but they still depend on motors. Kinesin and dynein are constantly transporting cargo up and down the axons. Neurotransmitter vesicles travel from the cell body to the synapse. Damaged organelles and misfolded proteins are transported back to the cell body for degradation. If the motors stop, the neuron starves at the synapse and accumulates garbage at the cell body. Axons degenerate. Neurons die. This is what happens in some neurodegenerative diseases. Mutations in motor proteins cause axonal transport to fail, leading to progressive loss of motor neurons or sensory neurons. Charcot-Marie-Tooth disease, hereditary spastic paraplegia, both caused by defects in kinesin or dynein. So motors aren't just for development, they're for life. Continuous, essential, irreplaceable, and all of it. Every sperm swim, every ciliary beat, every chromosome separation, every neuronal transport event traces back to the same molecular machinery. Dynein walking on microtubules, kinesin walking on microtubules, myosin pulling on actin, ATP broken down, energy released, conformational changes, force generated. Over and over and over, the motors that keep you alive are the same motors that kept your ancestors alive, and their ancestors, and theirs, an unbroken chain of motion, inherited across billions of years. So let's pull back, let's look at what we've traced. We started with a bacterium, spinning its flagellum at 100,000 RPM through water that behaves like molasses. We moved to protists, coordinating thousands of cilia without a brain. We followed sperm swimming through the human reproductive tract, powered by the same dynein motors that beat in algae. We saw how your lungs clear mucus, how your ears detect sound, how your cells divide, all through molecular motors that have been running continuously for over 3 billion years. And here's what stands out. None of this was designed, not in the way we design things. When humans build a motor, we start with a plan. We draw blueprints. We machine parts to precise specifications. We assemble them in a specific order, with tools and jigs and careful measurements. And we build in redundancy, fail-safes, maintenance schedules. We design from the top down. But molecular motors didn't come from a blueprint. They emerged, bottom up, from chemistry that learned to move, from proteins that folded into shapes that could capture energy from ATP and convert it into force, from random mutations that occasionally produced a protein that worked slightly better than the one before, from billions of years of selection, filtering out the failures, amplifying the successes, refining the mechanisms one small change at a time, and the result is machinery that by almost every metric exceeds what we can build. Efficiency. The bacterial flagellum motor converts nearly 100% of its input energy into mechanical work. ATP synthase operates at close to perfect thermodynamic efficiency. Human-engineered motors, electric, combustion, hydraulic, typically operate at 70 to 90% efficiency at best. We lose energy as heat, friction, vibration. Molecular motors don't. They're operating at the physical limits of what's possible. Precision? Kinesin steps along microtubules with 8 nanometer precision, placing each foot exactly on the next binding site, every time. Dynein coordinates thousands of motors across a cilium, producing smooth, rhythmic beats. The tolerances are atomic, there's no slop, no wobble, just perfect, repeatable motion. Durability? These cilia in your lungs beat 10 to 15 times per second, 24 hours a day, for your entire life. That's over 400 million beats per year. Multiply that by a 70-year lifespan, and you're looking at 30 billion beats. The motors don't wear out. They don't need oil changes. They don't need maintenance. They run until the cell dies, and when the cell is replaced, the new cell builds new motors and starts the cycle again. Self-repair. Molecular motors are constantly being degraded and rebuilt. Proteins are broken down by cellular machinery and recycled. New proteins are synthesized from amino acids, folded by chaperones, and assembled into functional motors. The motor you had yesterday isn't the same motor you have today at the molecular level, but the function is identical. The structure is preserved. It's ship of Theseus at the nanoscale. And self-assembly? Perhaps most remarkably, molecular motors build themselves. There's no external construction crew, no assembly line. The proteins are made in the cytoplasm. They diffuse through the crowded cellular environment, and they find each other. They recognize complementary surfaces, lock together, and form functional complexes. The bacterial flagellum assembles in about 20 minutes. The cilium assembles over a few hours. No instructions needed beyond the shapes of the proteins themselves. This is emergence. Complex, coordinated, functional structures arising from simple rules and local interactions. No top-down control, no global coordinator, just molecules, following the laws of chemistry and physics, and somehow producing motors that work. And this is what life does, not just with motors, but with everything. Metabolism, replication, sensing, responding. It's all emergent, all self-organized, all built from the bottom up, through interactions between molecules that have no knowledge of the larger structures they're building or the functions they're serving. The cell doesn't know it's alive. The motor protein doesn't know it's moving something. There's no intentionality at the molecular level, just chemistry. Bind ATP, change shape, release ADP, reset, repeat. Do these enough times, with enough proteins, and you get a flagellum that propels a bacterium toward food. You get cilia, that clear mucus from lungs. You get a heart that beats 100,000 times a day, pumping blood through 100,000 kilometers of vessels. Motion at every scale, built from motion at the molecular scale. And humans are trying to replicate this, to build synthetic molecular motors, to engineer molecules that can walk, rotate, pump, transport. There's been progress. In 2016, Jean-Pierre Sauvage, Fraser Stoddart, and Ben Feringa shared the Nobel Prize in Chemistry for their work on molecular machines. They built molecules with moving parts, rotors that spin when exposed to light, shuttles that slide along molecular tracks, rings that open and close. These are artificial molecular motors, chemically synthesized, designed by humans, but they're primitive compared to what biology has achieved. The synthetic motors are slower, less efficient. They require specific conditions, particular solvents, temperatures, light wavelengths. They don't self-assemble, they don't self-repair, and they can't coordinate with thousands of other motors to produce complex large-scale motion. We're learning, but we're far behind. Evolution had a 3 billion year head start. Still, the potential is enormous. If we could build molecular motors that rival biological ones, the applications would be transformative. Imagine drug delivery systems where molecular motors carry medicine directly to cancer cells, navigating through tissue, targeting with precision. Imagine nanoscale factories where motors assemble molecules one atom at a time, building materials with properties we can't achieve with bulk manufacturing. Imagine self-healing materials where motors repair damage as it occurs, reknitting broken bonds, restoring structure. Some of this is already happening. Scientists are using DNA nanotechnology to build molecular walkers, strands of DNA with legs that can step along DNA tracks, carrying cargo as they go. They're slow, and they only work in test tubes under carefully controlled conditions, but they work. They demonstrate the principle, and we're learning from biology. Researchers are studying dynein and kinesin, mapping their structures at atomic resolution, watching them step in real time, figuring out how the conformational changes translate into force. The goal is to understand the design principles, not to copy them exactly, but to extract the ideas and apply them to synthetic systems. Because molecular motors solve a problem that's fundamental to nanotechnology. How do you do work at the nanoscale? How do you move things, build things, manipulate things when everything is buffeted by Brownian motion and you can't use macroscopic tools? The answer is, You use the same strategy life uses. You harness energy from chemical reactions. You build structures that bias random motion. You create directional processes from reversible interactions. You let the system self-organize. And if we can do that, if we can build motors that self-assemble, self-repair and coordinate, we'll have crossed a threshold. We'll be able to build machines at the scale where biology operates. We'll be able to interface with living systems in ways that aren't possible with current technology. We'll be able to repair cells from the inside, to engineer tissues, to control biological processes at the molecular level. But even if we never achieve that, even if synthetic molecular motors remain a distant goal, what we've learned is profound. Because understanding how life moves, really understanding it, at the level of atoms and molecules and forces measured in piconewtons, reveals something fundamental about what life is. Life is not static. It's not passive. It's not an arrangement of molecules that happens to persist. Life is motion. Constant, directed, purposeful motion. From the bacterial flagellum spinning to the heart pumping, to neurons firing to muscles contracting. Motion is the signature of life. When motion stops, life ends. And that motion doesn't require complexity in the way we usually think about it. A bacterium has no brain, no organs, no differentiated tissues, but it moves with purpose, it navigates, it hunts, it escapes, all through molecular motors and chemical gradients and mechanical feedback. The intelligence, such as it is distributed, emergent, encoded in the structure and dynamics of the system, not in any central processor. And that's true at every scale. Your brain coordinates your movements, but your brain is itself built from cells that move, that transport, that communicate through motors and channels and signals. There's no homunculus, no ghost in the machine, just layers of motion, from molecules to cells to tissues to organs to organisms, motion all the way down, motion all the way up, and it's old. The motors in your cells are descended from motors that were spinning in ancient bacteria 3 billion years ago. They've been refined, adapted, duplicated, specialized, but they've never been replaced. Evolution found a solution that worked, and life has been building on it ever since. So when we ask, how did life learn to move before it had muscles? The answer is, it learned through trial and error over billions of years, through the same process that shapes everything in biology. Random mutation, natural selection, drift, occasional catastrophe, survival of whatever worked well enough to reproduce, and what worked was chemistry. Proteins that could change shape, membranes that could anchor rotating rings, filaments that could slide past each other, energy gradients that could be harnessed. Simple components interacting through simple rules, producing complex coordinated functional behavior. That's how life learned to move, not with blueprints or plans or foresight, but with molecules that stumbled, randomly, into configurations that generated motion. And once that motion existed, once cells could swim and hunt and escape, the rest followed. The evolutionary arms race, the diversification, the scaling up from single cells to multicellular organisms to animals with nervous systems and muscles and bones. But the motors never changed. They're still there, still running, in bacteria, in protists, in algae, in the cells of your lungs and your ears and your reproductive tract. Ancient, conserved, universal. And every time you breathe, every time you hear a sound, every time your heart beats, you're running on engines that are older than bones, older than eyes, older than brains, engines that existed before anything we'd recognize as an animal. Engines built from nothing but folded proteins and chemical energy. That's the legacy we inherited. Not just genes, not just DNA, but machinery. Working machinery. Machinery that moves us, heals us, senses for us, builds us. Machinery that connects us to the first cells that ever moved in ancient oceans, under a sky with no oxygen, on a planet that looked nothing like the one we live on now. The motors are still running, and they'll keep running, passed down through every generation until there are no more generations to pass them to. Motion is life, and life has been moving for 3 billion years.